Hi, everybody. He did a great job of introducing. I'm standing up here. I want to make certain I say at the beginning that I represent a much larger team that spans both the Charles River campus, the medical campus, as well as outside collaborators. Dr. Hamer did an excellent job of introducing MLS. He also did a great job of introducing the concept of genomics. And I think the main thing I want to get across today is that we have had, over the past 5 years, we have had a complete sea change in how people understand infectious disease from the genomics standpoint. Prior to about 2015, the idea of understanding the full genome was a nice to have, was something that you would get after a while. In the intervening time period, we have had major outbreaks of things like Ebola and then Zika that have changed how people think about individual infectious disease genomes. And the question is not, can we have one, please? Can we understand just one? It's, can we understand the latest one that just occurred as quickly as possible? You guys know this because the idea of variants in COVID-19 is on everybody's mind. It's in the newspaper above the fold on the first page. So this has been an incredible sea change, and it has made a huge difference. My lab participates in this as part of a larger group, and really what this has evolved to is that we can get information on a time scale where we can really inform an understanding of disease transmission based on the genome. If you think of traditional contact tracing, what I'm showing you here is someone who has tested positive, and this is somebody interviewing them. They have a bunch of questions. That person gives answers. And from that, the contact tracer, the epidemiologist can get a pretty good idea of things, of transmission chains. But there are often big holes. And one of the problems is this individual may have a reason to not tell the entire truth, may not actually remember things. So what we're doing in genomic contact tracing is we're sequencing the genome, and the genome has a much harder time lying. The genome has a much harder time forgetting things. And so what it allows is for us to use the identity of that genome. We may not know everything about this individual, but we know where that genome's neighbors or identical twins have been through looking elsewhere. And so we can use that to help contact tracers not only confirm, yes, the connections we thought were correct are actually correct. These individuals have exactly the same viral genome. We can also add people. We can add clusters that were previously not appreciated, or integrate more people into a cluster to allow for better understanding of what's going on. The reason this is really helpful is it allows people now to say, we understand whether how the transmission happened. We also understand how transmission didn't happen. If we have a quarantine time period and we let people out, does that do the National Emerging Infectious Diseases Labs, which is depicted here? And essentially what we are doing at this point is two different things. I told you about genome sequencing. That allows us to identify variants. It allows us to do genetic ID tracking. And when I say we, I actually largely mean one person. Jacqueline Trosinovich is in the back. Jackie, if you could stand up just briefly. This is the engine that has driven all of the genomic sequencing that's going on. She's a graduate student in bioinformatics. And I asked her to come. I didn't tell her I was going to embarrass the living daylights out of her, because you don't tell people that. And this has been immensely useful. One of the other things that this has also given us, because of the power of the needle to do research at multiple biosafety levels, is we can also use some of these variants that are of interest for research purposes and immediately culture them and share them not only within this building, but also around Boston and around the nation, to understand how these variants are changing functions. So we can meld not only contact tracing, but also what we're doing. This is just to say that it's a village. I think I heard a beep saying I'm done. And this is to say there's a lot of places in which this can be useful. We're happy to work with people. And I'll stop there.